So my name is Guy. I'm the Ensembl Plants project leader. I've been here for a while, about 7 years, and I'm going to talk about how we can explore plant genes and genomes with Ensembl. So the way this is going to be, I'm going to give a brief overview of what we do in EBI, what Ensembl Plants is, and sort of go a bit into Ensembl Plants out of this brief whistle-stop tour. Pan-genomes in Ensembl Plants, and there's a nice pan-genomics talk in a few weeks, so it should also be sort of a nice thing to know about. How we get new species into Ensembl. It might be interesting for you if there's a specific species which you would like to see in Ensembl. What happens if you want to get bulk data, and also resilient species and orphan crops, and finally a bit about the Darwin tree of life. So what do we do in EBI? So we do quite a bit of things, and one thing we do is we contribute, we do research. It's not the main thing we do in EBI, but it's one of the things. We're part of Elixir. We do training like we're doing right now, but I think the thing we mostly do in EBI, the thing we do in Ensembl, is we provide freely available data and bioinformatics services to the scientific community, and you'll soon find out how. We're located in 6 different sites all over Europe. EMBL-EBI Kingston, United Kingdom. This is where EBI is. Then you have Heidelberg, which is sort of like the bigger site, but also sites in Germany, France, and Italy. And Spain, sorry. And this is Ensembl Plants. This is what I'm going to be talking about for the next half hour or so. And if you sort of ask what Ensembl Plants is, so this sort of short answer is it's an open access platform for genomic data and annotation. What does that mean? We'll find out in a bit. It's a fairly stable project. It's been around for over 20 years, since about 2000, was sort of the human genome was sequenced, and Ensembl Plants was added in 2009 as part of what is called Ensembl Genomes. Everything is completely free and open source, which means a lot of our sort of code and our data is online. You can download it, and there's no secrecy. You don't have sort of need to sign up for anything or get a confirmation email or do a username or password. You can just go in and start playing around. And if I try to sort of explain what Ensembl Plants is, so a nice metaphor is like this big machine with all sort of these cogs and wheels going around, and some of the things I'm going to talk about today, and there's many others, is the genome browser, which is sort of a way to sort of look at genomes, the variation data which we have, comparative genomics, which I'll show later on, how to sort of get data in bulk. It's also a big part of the EBI sort of tool, so it connects sort of other EBI things. I'll talk about that a bit later on. Training, which I'm doing now, release cycles, which I'll show in a bit. APIs, and also the variant effect predictor, and I could just keep on doing all these little things here if this was a bit of a longer talk, but in the end I'll also explain how you can learn more about Ensembl, so that should be interesting. And we have a release cycle. We release sort of a new version of Ensembl, or a new release of Ensembl about every 3 or 4 months, sort of 3 or 4 times a year. And this includes new genome assemblies which we add. If we need to do software updates, we do this comparative analysis run on all the genomes. We update variation and regulation data. We also do updated interfaces. So this means once something gets into Ensembl, it takes a while for it to go out. That being said, we also have something fairly new. This is Ensembl Rapid Release site. You can just sort of look for Ensembl Rapid Release and find it. And this is, like it sounds, much more rapid. There's a turnover about every 2 weeks for every release. However, most of the plant data is still in sort of the main Ensembl Plants. Also in the main Ensembl Plants we have a bit more of comparative analysis, so I'm going to talk more about that. But this is something to know, and in the future more things will go into that. For example, the Darwin tree of life, which I'll show later. So we have quite a bit of genomes, over 120 genomes, and these are a few of them. So if you don't see your genome of choice, hopefully it's in one of the others, 125. That being said, the genome we've been sort of working on quite a bit lately is wheat, just because we had funding to work on wheat, and it's sort of our #1 search species. Also, a few years ago, in 2018, there was this really game changer when the assembly of wheat was finally sequenced, which was really awesome, and Ensembl was the first place where it was publicly available. So Quite a few of my examples will be from the world of wheat, but if you're not working on wheat, it looks very much the same in all of the other sort of genomes in Ensembl. And you'll see I also show a few other besides wheat, so don't worry if you're not a wheat person. So this is Ensembl Plants. This is how it actually looks, and this is sort of the wheat page. Again, it looks very similar in other pages. And every time sort of I see this new bioinformatics resource, the first thing I like to sort of do is just click on the examples, just have a sort of feel of the site, see what's happening. So that's what I'm going to do with you for the next 5 or so minutes, and we'll start with the example region. And this brings you to Ensembl Plants genome browser. And this is sort of like the heart of Ensembl. This is like where everything happens. You find your chromosome of choice, you sort of zoom in, see what you want, and then you have a lot of information about genes. You can see what the exons are, the introns, genes from different sources in different colors, variation data, which I will talk about in a bit, non-coding exons. And the way it actually looks in Ensembl, you have sort of this higher pane, this very high level one, and then the lower pane is sort of what I just showed you. And you can really sort of zoom in, which is nice, actually see the DNA. And also sort of zoom out for a high level, more of a high level picture. You can go left, you can go right. And if you're here, sort of the natural thing to do was to just click on a gene and see what happens. So this is what happens. You get to sort of this gene page where you have a bit of information about this gene, orthologs, paralogs, transcripts, and what this gene is. This is a heat shock protein, sort of another mini genome browser. And the next step you would probably do is click on transcript, which brings you to the transcript page. Here you have again sort of where it's located, and you can also scroll back there. This transcript has 7 exons. You can get domains and features, variation, also link to Uniport, external links. So I've talked about how sort of a lot of the EBI projects are sort of connected. So this is sort of the nice connection from here. And the next step, you're already in the transcript. Let's look at the sequence. You can sort of click on exon, but also on cDNA and protein. So this sort of gives you the exon page. And you can see the different exons. In this case, you also have the variation in the sequence, which is really nice. So this was very, very quick, sort of a whistle-stop tour of how to sort of start from the genome browser, chromosome level, then go to the gene level, then go to the transcript and sequence level. And what I'm sort of trying to show you here, I hope, is that getting to really interesting stuff in Ensembl is fairly quick and easy, and hopefully very intuitive. And there's so many other things sort of you can do on the way and learn and turn off features and sort of do a lot of really interesting and cool things. But a lot of the important stuff is easy, and I hope sort of that's something that you will find helpful. Another thing which was recently added into Ensembl is AlphaFold. Basically, if you've heard about it, it's a way to see structure prediction, and we don't have that for all the species. This is an example from corn, from Z-maize, and this is so if you just click on the left, this is how it looks, which is really nice. In genomes, when we also have variation, you can also see variation there, which is really nice. And like I said, we only have this for a handful of species, but in the next release, in a few months, we're going to have that for pretty much everything, which is really cool. So we looked at the example region, now let's look at the example gene tree. So this is what we actually do every release. More or less is sort of the big picture, and there's a link here if you want to know more. I know this will be available via PDF later on. So we just do basically blast all versus all. We get these clusters. We use a tree algorithm to get the trees. We get orthologs and paralogs. And to show an example, so let's move from wheat into rice. And if you look at this specific gene called DRAW1, you can see that it's connected to yield under drought. And sadly, you know, with climate change and everything, this is becoming more of a more important thing. I'll talk a bit about climate change in the future and what we sort of need to do. But if this is sort of a gene that you're interested in looking at, you can just search for it like we did before in wheat, and then so this is the same gene page that we just saw before. And here on the left you have plant comparative gene tree, and this sort of brings you to the comparative gene tree. And if we sort of look a bit closer, you can see this is DRAW1 that we looked at, but then you see sort of its closest orthologs. So Oryza, Glabirema, That's African rice, and you also have a few other sort of wild rice relatives, Oryza indica. So this is obviously very useful information, because then you want to sort of look at the different orthologs and the genes and sort of see how similar they are, how different, and this is a really nice way to sort of go and start researching this. And another way is just to look at ortholog view. So here's sort of the list of different orthologs. You can see we also have gene under conservation score, which sort of means how many neighboring genes are also orthologs. Whole genome alignment as well. And again, another sort of useful tool to sort of search this gene, and then you can just, let's say, click on one of these genes and you see the same gene page which we saw, but let's say, for example, for Agilopstachy and so on. And finally, let me just talk to you a bit about variants. So here we're actually jumping to the world of common bean, Phaseros vulgaris. And here you have a lot of information about this specific variant. You see what its most of the consequences. This is sort of the variant effect predictor. This is a framesheet variant. There are alleles, where it's located. And if you click that, you sort of get to the genome browser, which we saw before, a few other external links. And if you sort of look at the sample genotypes, you can see how it looks in all the different samples. So if you go back to sort of the genome browser view, which we saw, you can see there's sort of all these variants. And just clicking one of them will bring you back to the variant page. Another way to see it is through sort of this more visual display. And here you can see the red ones are the ones that are stop codons. In this case, these are the ones that you might find interesting. And clicking them, you get a bit more information about this specific variant. And if you click that again, you get sort of to the page that we saw before. So everything is sort of interconnected. You can go back, you can go forward, you can try different things. Another nice thing about the variation data that we can import it from the European Variation Archive. Here you see sort of variation sets for common bean, and this is sort of fairly easy for us to do. So if you know about the variation data, that is useful, and you can just sort of, or if you work in variation data, just getting into EBI is fairly easy, and then just getting into Ensembl is also really good, and then you can sort of go and study it. So this was quite a bit about genomics and what we do and what we show. And I'm going to sort of jump to a bit of a different topic here just for one slide, and this is about breeding. And I know you're going to learn a bit more about breeding with the pangenomics talk in a few weeks. But just to say that creating new varieties, and this is wheat, but it's also fairly similar to other crops, is long and complicated. You have to sort of do screening and then testing and then more testing and then market. And if you sort of look at all this F1, F2, F3, each stage is actually a year. So getting a new variety can take 10 to 11 years. And all the genomics information and data we have, which is fairly new and sort of a real revolution, it's not a magic bullet. You still have to go to the field, you still have to do all this testing and so on. But it helps. It makes a difference. Sort of actually being to look at the actual genome and actually look at the gene, I think is really, in a way, it is a game changer. It's not a magic bullet, but it does change the field. So it's really useful to have this, and as more and more genomes sort of being sequenced, I think that's something that can really help us. And again, talking about climate change, trying to find better varieties in the future, more resilient ones, ones that are sort of need less water. I think that's a very important thing, and the fact that we can help this with tools like Ensembl and others, I think is great. Okay, so this was EBI Ensembl Plants, sort of a very brief tour of Ensembl. Let's talk about pangenomes a bit. So I'm not going to go too much into what a pangenome is, and again, if you join the talk about pangenomics, I think that will give a bit more information. I'm going to talk about this project we did in the wheat pangenome, and this was published in Nature just over a year ago. And if you go back into the wheat page, you can see you have this cultivar view in Ensembl Plants, and if you click that, you get sort of this list of wheat cultivars or wheat varieties. So if in the past it took quite a while to get the first wheat sequence, then this consortium did another 14 wheat sequences, which is really cool. And if you click on one of them, you just get to the same genome browser page that we saw just before. Another nice thing is that we have this wheat-specific gene tree. So this is a gene tree just for wheat. So sort of the wheat and wheat cultivars and also other wheat wild relatives. This is another useful resource and something we just added recently and very useful to the wheat community. We're also working on other pangenomes. So there's also a rice pangenome. It's called Magic 16, rice varieties and relatives. And we're also doing a gene tree multiple alignment plan in the future. And this is part of the Panorizer project. And there's more and more pangenomes coming out, which is really a fantastic challenge. Something for us to sort of see how we can scale, how we can add. I know there's a oat pangenome with 30 pangenomes. There's a barley pangenome with about 70 genomes. So getting the onto Ensembl is quite a challenge. And also the next challenge is sort of how to use this, because as scientists, as breeders, we're using to work with a reference, and suddenly you have 10, 20, 50, 70, 100 genomes, which is an extraordinary amount of information, but what's sort of the best way to visualize it? What's the best way to use it for breeding? And again, you'll perhaps get some answers in a few weeks, but this is a new thing in the world of genomics, this pangenomes, especially this plant pangenomes. I'd say just something that happened in the last 3 or 4 years, and we're still learning as bioinformaticians and as scientists how to use them, and I think this is really interesting to find out what's going to happen there. And just to show that we have the rice pangenome in the rapid release site, and that's going to go into the main site fairly soon. And here's what I want to talk about, a bit about getting new species into Ensembl. So how do we find new plant species? And again, might be that species that you're interested is in Ensembl, but it might be that it isn't. And then this is sort of something interesting for you, and something I can also encourage you to sort of ask questions and see how we can sort of start this. So for sort of major crops, model organisms, we usually work with a community, for example, wheat, rice, and so on. But there's also a huge variety of plants outside of the crop world, a huge variety of plant institutes. And what we mostly do is sort of just try to read the literature, see if there's a new plant species, a new plant genomes coming out. And here we have quite a big challenge, which I just want to talk briefly about it. That most of these plant, well, not most, but many of these plant assemblies that are published are not submitted to the INSDC public archives. And this includes the ENA in EBI, GeneBank in NCPI, and DDBJ, International Institute of Genetics. And there's quite a lot of advantages of submitting to the archives. Basically, the assembly will be in a stable place, will be available for years to come. This is, again, part of, well, ENA is part of EBI, but also part of INSDC. We know this is not going anywhere. It's also quality test. But more importantly for us, we're only allowed to display genomes that are in these archives, according to the browser agreement which we have. And just to give a quick example, that's not always the case. And this is sort of a collection of horticultural plant genomes, which I found a few years ago, and I was super excited. This is a lot of really fantastic assemblies. Let's add them to Ensembl. But sadly, only one of them was in the public archives in the case of asparagus, which is a bit of a shame, because obviously for us in Ensembl we'd like to have them, but also for the communities that might be using them. I think having them in genomic resources like Ensembl could be very useful. So this is something we're trying to sort of work with the community, and every time I give a talk, try to explain how important this is. So, are you working on a genome? Are you looking in a genome? It's not in Ensembl Plants. So what do you need to do it? Obviously, it needs to be in the public archives, as I just said. It does require full genome annotation. That doesn't need to be in the archives. We very much like to work with the community when updating a genome, because then we know which genome the community is using. Sometimes there's a few of them, and then if there's any issues, if we're not sure where the genes are, if there's naming problems, if we want to get a bit more scientific data to sort of display, that's really good for us. So we usually prioritize that when it comes to the community. And if you're interested, so get in touch with helpdesk at ensembl.org. And they're actually here, the outreach team, which I'm going to talk about in a bit. Or you can also just email me, and it's usually very easy to find me. Just Google Guy EBI and you should see my face. Let's talk a bit about bulk data. So I've sort of been showing you how Ensembl looks on the genome browser, but sometimes you just need a lot of data. And just going, I don't know, if you want sequences, just going gene by gene and downloading it is obviously not enough. One thing I haven't talked about is Biomart, and you can learn more about that and explain it away, but there's also other ways, and some do require programmatic skills. So if you're sort of into programming, that can be very useful for you. And the good thing is there's plenty of examples and documentation. And there's this book chapter which we published last year about sort of how to get that. And just to say that in GitHub, in Ensembl Plant scripts, that's also in the book chapter, you have a lot of sort of recipes, examples, how to get that in sort of different languages, in Perl, in Python, in R. And we also have something called the Ensembl RESTful API, which is a way to sort of get things in a sort of faster way. And here's sort of an example of just very simple code you can use and then just get information. And the nice thing is you can do it in very different languages. You can do it in Perl, in Python, in Ruby, and so on. Let's talk about resilient species and orphan crops. So, diversity is important, obviously, in everything, but also in plants. And what's been happening in the past, I don't know, thousands of years, a lot of commercial crops, or sort of the more widely used crops, lose genetic diversity because of domestication. Basically, you try to grow the same ones over and over again, and you sort of inbreed them. And they lose the diversity. And what we sort of want to look at, what we want to explore as scientists, as breeders, is sort of this wild, non-domesticated species and model species, also others that are important in ecological studies. And the interesting is, the more diversity we have, the higher the chances of we get this functional annotation from one species to another. So if you find something in wild wheat relative, then you can sort of try and see how you can move it into wheat. And there's a lot of advantages of having access to these genomes in tools like Ensembl or others. And just a few examples of ones we've recently added. This is fonio millet, and this is a very fast maturing cereal, and it's often grown to avoid food shortage during the lean season. Another one is rye, which is sort of very resistant to many fungal diseases. And cowpea, it's again very resilient in harsh conditions, including hot and dry environments. So getting this sort of species, sort of very different, very resilient species into Ensembl, I think, is something very useful. And then again, talking to the scientific community about them. Another example is teff, which we recently added. And here it's sort of what I'm saying before, that you have about 30 crop species and do about 9% of the world's food needs, but they're very much, but then the diversity is very narrowed. And then you have something called orphan crops. They're called orphan crops because they're not as well funded, they're not as well researched, they're underutilized, as I said, but then they have a lot of really desirable nutritional profiles and a lot of untapped genetic potential. So sequencing these crops, getting them into sort of the archives and into tools like Ensembl, I think is something that can be very useful for the community. And this is something we're really trying to do, trying to sort of look out for these orphan crops. And the good news is, basically it's getting easier and cheaper to get them. So a lot of them sort of have been recently sequenced, or they've been sequenced very well. A few that we have is cassava, pigeon pea, and grass pea, which I'm going to talk about in a bit, which is still going to be added. And again, if this is a genome in the public archives as described, we're very happy to add it in Ensembl. So if you're working on a orphan crop, you're interested in orphan crop and it's not in Ensembl, get in touch with us and we'll be happy to work on it. And just showing an example of grass pea. So grass pea is a very hardy plant. Often it's the last crop standing. It can withstand extreme environments. So brilliant, why don't we all eat grass pea? But unfortunately, it can also be poisonous. So if you eat a bit of grass pea, that's fine. If you eat only grass pea, which sadly is the case in cases of starvation, you can also get paralyzed, which is fairly horrible. But this is exactly where we can make a difference. So once we have this genome sequence, once we do have it available to the community, we can sort of try to understand why this happens and maybe find the specific genes which are poisonous and sort of try to change that. So just sort of highlighting the importance of getting these orphan crops sequenced and sort of in public bioinformatics tools. We're also working on the Darwin Tree of Life, and this is sequencing all of the eukaryotic species organisms in Britain and Ireland, so quite a few of them are plants, and this will go into the rapid release site, which I talked about just before. And in this case, we'll do our own annotation as compared to community annotation. Another one is the African Genome Project, which will also get into Ensembl eventually. I know there's also a talk about the Q tree of life, so a lot of there's this big, massive projects of sequencing, and hopefully all of them will eventually get into Ensembl, so in a much bigger scale. Another thing we're working on is a new version of Ensembl. It's called Ensembl 2020, but 2020 has gone already, as we all know. But this sort of a first version of this will be available by the end of the year, and then as we go on, this will sort of become bigger. And this is just, and first thing we'll show is that Ensembl Plants will be part of the bigger Ensembl, and this will sort of be easier to use, faster and quicker. That being said, the Ensembl, you all know, now isn't going anywhere. It's going to be around for quite a few years, even while this is up. So we're not going to just turn it off immediately. So it's still going to be on for a while. So it's a lot of information, fairly quickly, lots of sort of different things that I talked about. But the question is, if you would like to know more about Ensembl, sort of dive a bit deeper on how to use it, how to use Biomart, how to sort of download things, learn a bit more about the API. We have a fantastic outreach team, and they're actually here today on this webinar, so you can ask them questions, and they give workshops and courses for organizations and institutions. Online, it's always free. Face-to-face for the cost of travel, so if you're located in the UK, that's fairly cheap. Europe also not too expensive. If it's other places in the world, obviously a bit more complicated, but there are options, so do talk to us. And you can just email helpdesk@ensemblgenomes.org. That's also for Ensembl issues. If there's something wrong, if you have any questions, and they answer fairly quickly. Also, if you're interested, just drop me an email, guy@EBI. So this is a big thank you to all the sort of different collaborators from different projects, from Panorama, from Designing Future with, which we're part of, our collaborators Min, Elixir, and what you can also see here is that Ensembl Plants, there's not many of us. So there's Sarah, who's sort of the team leader, also leading the outreach team, which we saw before. There's Shraddha, who's working on rice and wheat and a few other projects, and me, that I'm leading the project. And the fact that we sort of managed to maintain this fairly big machine with all of its different things is that we have quite a lot of people also in Ensembl. So not all of these are Ensembl, sort of like the main section, many of them are. So the variation team and sort of the people who do it web and the production and outreach and comparative analysis and quite a few others. So just a big thanks to all my colleagues, and very happy to take a few questions.
There are some open questions right now in the Q&A box. So I'll read that out, Guy, for you. So one is, do autotetraploids generally have more resilient traits?
I'm not exactly sure. I don't think that's necessary. I guess it also depends very much on how much sort of they've been breeded. So in many cases, you might have tetraploids or hexaploids like wheat that sort of they've been breeded for yield, but not necessarily are resilient. But then you would have sort of more wild relatives that perhaps are, they're not as good for yield, they might not be as nutritious, but they're just more they just survive better. A good case sort of is graspy, which I don't think is an tetraploid. So I don't think that's necessarily the case.
Thank you. Next question is, what is the basis of low and high confidence genes in Ensembl gene annotation pipeline?
Okay, that's a really good question. And for wheat and for a few others, we do have high and low confidence genes. So this is actually what we get from the community. So when the wheat community or the International Wheat Consortium did the wheat genome, they decided which is high and low confidence. And this is depending on a number of factors. And every time it's a bit different. But if you have expression evidence for a gene, that would sort of make it a bit more high confident, or if it has very strong orthologs, in other genomes that will usually be high confident, and low ones are ones that are perhaps just prediction but no other evidence. But if you're sort of interested in a specific genome, I would sort of encourage us to go and look at the paper, for example, the wheat paper, and see exactly what the differences are. But those are like the main things.
Okay, so next question is, how would you compare Ensembl to other plant platforms like Phytosome or Plaza, etc.?
Okay, so I guess every platform is a bit different, and I think there are things we have that Phytosome doesn't, and likewise, I think Phytosome have. I'm not sure if they need things to be in the public archive, so they have a lot of much bigger list. I think a lot of us, I think we're more based on wheat than they are. But yeah, I think everyone has advantages and disadvantages in actually having this different genomes, different platforms, and then having deal with all different platforms. I think is something that can be sort of really useful for the community. And if you're using a few of them, you can compare and see which one is better for you and which one is useful for you. And I think it's really great that we have a sort of these different genomic resources like Ensembl and Phytosome and Plaza and also Grain Genes and a few others. But I don't think there's a very quick answer what the exact difference is. So there was a question sort of that disappeared about, I think it was peanut, getting peanut into Ensembl Plants, so that's something we're in contact with the peanut community, and hopefully there's a new one that's coming out, so hopefully once that genome is sequenced and is in the public archives, we'll be adding that into Ensembl. And do please, if you see your favorite genome and it's on Ensembl, just let us know, and once we sort of get things from the community, I think we try to prioritize that.
Okay, there's one question which just came in. I want to work on genetic similarities between plants and other symbiotic organisms or their parasites such as bacteria, fungi, insects, etc. I'm interested in finding the relationship between plants families and the horizontal transfer of genes by the mentioned organisms. But I have no experience in genetic work, but I'm very interested in doing it. Do you have information about how it works and the right software?
So I don't know how much I can help you, but one thing I can sort of say, and maybe it's a shame that I didn't show it in my talk, that we also have Ensembl bacteria and Ensembl fungi, so it's basically the same thing for bacteria and fungi, and then sort of you can go and explore that. We also have quite a few of insects in Ensembl. So I just showed you the plants one, but a lot of these other sort of divisions are also in Ensembl, so that could be sort of a good starting point, going and looking them and trying to understand a bit more.